Misty, thanks for joining us today.
Thank you for having me.
Certainly. So I know that you just came fresh from a poster that you just presented, so this is a perfect time to talk about that.
Yes.
And wanted to, just to start off, if you want to just give us a general idea of the poster, and we'll start there.
So last year I received the Epilepsy Foundation TRICE Award, the Targeted Research Initiative for Cannabidiol and Epilepsy, to look at the interactions of cannabidiol with other anticonvulsant drugs. So in my study, I actually looked at 5 mechanistically different anticonvulsant drugs and used isobolographic analysis to see how those drugs interacted in the 6 Hertz limbic model of epilepsy. And we actually had some really interesting findings.
Yeah, no, very good. Well, so good. Well, I'll tell you what, let's do this. Do you want to you were just talking about the most frequently asked questions. So do you want to, why don't you start with that and we'll go from there.
So most people were curious, what exactly is an isobologram study? And it's actually a fancy way of saying it's a combination study. So what we did was we took cannabidiol and the other 5 drugs, which include clobazam, levetiracetam, lacosamide, carbamazepine and valproate, sorry about that. And we determined their effective doses, their median effective doses, or ED50s. And then we were actually able to use ratios, fixed ratios of those ED50s in combination to determine how well the drugs work together. If the drugs didn't interact at all, then they had just an additive interaction. They didn't hurt or hinder one another. A synergistic interaction actually made the combination work better than either drug alone, and an antagonistic interaction made the drugs actually hinder each other, so that you actually needed more drug to have the same effect as either drug alone. So we had some surprises and some exciting results. The most exciting was we found a synergistic interaction at the 1-to-1 fixed ratio combination of cannabidiol with levetiracetam. So it was very efficacious and really didn't have any toxicity associated.
Yeah, well, very good. So that was the, would you say that was the biggest finding of the combinations that you thought? Okay.
Yes.
Was that surprising? Was it something you expected,
well, the data was always surprising because we didn't really know what interactions to expect going in. But you know, in addition to looking at the anticonvulsant effects, we also were very interested to look in models of the comorbidities of epilepsy, including anxiety and depression. And we didn't find any indication to suggest that it helped or hindered animal behavior in those models, other than a little reduction in some of the hyperlocomotion, or I'm sorry, the locomotion of the animals in the tests that we performed. So all in all, the drugs were relatively safe at the doses that were efficacious in the model.
Yeah, well, very good. Did you have a way to think about it? I understand this is new data, so you know, if you're doing good research, you don't know what to expect, right?
Yes,
because it's a new thing. So I understand that side of it. But did you have a target dose for the CBD? Did you have a range of where you thought that should go? Actually,
the way we started the study was we determined those median effective doses for each drug, and that was our starting point. Because when you do the isobologram, all the combinations are based on ratios of that ED50 dose. So, for example, a 1-to-3 combination is actually 25% of the first drug and 75% of the second drug. A 1-to-1 combination is 50 and 50, and a 3-to-1 is 75-25, so that in combination they equal 1. So by giving those fixed ratio combinations, you can predict if it's strictly additive, if they're not interacting at all, how the drug should behave, and compare it statistically with what you actually get. So it's a model that's been used for some time, and it's highly predictive of drug interactions.
Yeah, oh, very good. Very good. And so that was a part of the study then to determine that dose for CBD?
Absolutely, absolutely. So we determined that cannabidiol and all of the other 5 drugs were all efficacious in the 6 Hertz model. We got the median effective doses for each of those drugs, the times of peak effect, where they had their maximum activity, and we used all of that information when we did the combination studies to get the best possible results.
Yeah, that's good.
Yep.
Well, good. So you had so levetiracetam and CBD seemed like a good combination.
They were our best.
All right. Yep. Any combinations we might want to avoid?
Sadly, there were.
Yeah.
Sadly, we had more of those than the good ones. So particularly carbamazepine didn't fare well. In 2 of the fixed ratios that we tested, there were significant antagonistic interactions. And simply what that means is you needed higher doses of each of the compounds to reach the same level of efficacy of the drugs by themselves. So that is something to watch for, and that really the take home for all of this research is that it's critical that we understand how the drugs are interacting with one another, because the patients who are most likely to be using cannabidiol are the patients who are using a lot of other anticonvulsant drugs, and really safety is a very high priority, and until we understand those interactions, really more research needs to be done.
Right. Well, it sounds like a good start, though.
Yep. It is.
And then again, so that's efficacy, and then anything on the side effect profile that stood out.
Well, it stood out in the fact that all of the drugs were efficacious at doses that were not toxic. Yeah. So at the doses that we determined, we saw very good efficacy and no real side effects to speak of.
That's good. And there wasn't one combination that stood out, like the carbamazepine and CBD together. Was that a trouble?
No.
So nothing came out on that side of it. It was just that they might not have worked as well synergistically.
Exactly.
Was probably the major issue.
Exactly. It did not. None of the combinations, even the antagonistic combinations, caused any overt toxicity.
Right. All right.
Yep.
Well, good enough. Fair enough. And then I guess this is a little. I know this is a tougher question, but you can at least comment for the audience.
Sure.
So the mechanisms. Any ideas about CBD and kind of how it may work from your perspective? And did that make any sense with what we know about the other medicines that are out there?
Well, cannabidiol is complicated. It has a number of different mechanisms that have been proposed. The majority of folks are interested in the potentiation of adenosine and the increase in adenosine and its inhibitory neurotransmission. But there are many, many other mechanisms that have been proposed, not only in epilepsy, but in a number of other fields where we can garner a lot of information and bring to the epilepsy community. We know that levetiracetam works through the SV2A presynaptic calcium receptor, and I can't really say that we can predict mechanistically what's going on there with the interaction. I will say that we have collected plasma and we have collected brain tissue from each of the animals that we tested, and we are in the process of analyzing the plasma levels of each of the drugs and the combinations to see if there's pharmacokinetic interactions. And that will really tell us a lot about the nature of the interactions we observed.
Yeah. That's great. Well, sounds like a great study.
It is.
Yeah.
And I have to, I would be remiss if I didn't say a huge thanks to the Epilepsy Foundation and the Epilepsy Therapy Project for making this research possible, and just encouraging everyone to support the basic research in epilepsy moving forward.
Well, thanks for being with us, and I would just echo that we need to know the basics before we can take the next step.
Absolutely.
And certainly in this issue, the more basics we know, the better off we're going to be.
Absolutely.
So,
Well, great. Well, thank you for having me.
Yes, well, thanks for joining today.
Thanks. Nice to meet you.